South America. I think South America. Good answer. Good answer.
Okay. Part 3 of Chapter 15. There's 4 parts to Chapter 15, so
Oh, really?
we've been talking about how information flows inside an organism from DNA to RNA to proteins. Yesterday we talked about how DNA turns into RNA through the process of transcription. We talked about ribosomes and how ribosomes are different in prokaryotic and eukaryotic. How many ribosomes? Well, no. No, that's not what I was going to walk in here. Never mind.
I don't know where he was here for knowledgeable.
Oh, he left the parking lot when I pulled in.
Oh my god.
Bertram?
Yeah.
He was here for knowledgeable this morning.
Yeah.
He probably had to go get something to drink.
Oh. Okay. So ribosomes. We're going to talk about ribosomes today. I meant to say RNA polymerase. How many different RNA polymerases are in eukaryotes? 3. So we have RNA polymerase 1, which makes what?
rRNA.
rRNA. And then we have RNA polymerase 2, which makes what? mRNA. And then RNA polymerase 3, which makes tRNAs. Okay? So we're going to talk about the ribosomal rRNAs and the tRNAs that are made, and then talk about how they participate in translation.
Does that mean we're ahead of schedule since it says January?
Yep, which is good
Good
because
deal.
we'll have we have to do more labs after Christmas. So. Okay, it's freaking out on me. Okay, translation objectives. So we'll talk about tRNAs, their structures, as well as ribosome structures, and then what they do. Understand how tRNA binds to an amino acid. And then we'll break down translation into its 3 steps: initiation, elongation, termination. Notice that there are some more differences between prokaryotes and eukaryotes. And then describe how some proteins are targeted to other parts of the cell. So like how they go to the ER and then from the ER to different parts of the cell. So the structure of tRNAs and rRNAs. We'll talk about tRNAs, which stands for transfer RNAs, and their main function is to carry amino acids to the mRNA and to the ribosome. We call it an adapter molecule because it's the link between the mRNA and the amino acid. It's bifunctional because one part of the tRNA interacts with the amino acid and another part interacts with the mRNA. So it's bifunctional, an adapter molecule that connects the link between those two. So before we get too far, remember we talked about protein structures back in Chapter 2, where primary structure is a unique set of amino acid sequences. Secondary structure is all about hydrogen bonds and how they form beta sheets and alpha helixes. Tertiary structure is like a combination of beta sheets and alpha helices, like helix turn helix or something like that, or beta barrow sheets. And then we have our quaternary structure, which is a combination of many polypeptides linked together to form a very complex protein. So if I show you this structure right here of a tRNA, how many different levels of protein structure can you see just from this diagram right here?
3.
Which ones?
The green, the blue, and the red.
Oh, I see how you got 3. I was gonna say you're correct, and now you're incorrect.
Oh.
Okay. So what's primary level about in proteins?
Amino acid sequence.
So do we have an amino acid sequence?
Yes.
Oh yeah. Okay. These all code for specific amino acids. So we definitely have primary. Notice we see some lines here. Those are hydrogen bonds. So we have secondary. We have secondary. And now the question is, do we
I think
have?
it's quaternary.
Well, we have 3, but it's tertiary.
50-50 chance.
Yeah. So this clover leaf shape actually kind of folds into an L shape when it's all said and done. And it's got some functional ends. So like down here, we have something called an anticodon. This is the part that will interact with the mRNA. At the top, we have this acceptor stem. Notice we have an OH group. Remember how you form bonds? Basically make water. Okay. So like a hydrogen will fuse with the OH, cleave it off, you form water, now you formed a bond. So there's that OH group, the acceptor stem, this is where the amino acid will link onto. So this is why it's an adapter. You can read the mRNA on one side, and then it has the the amino acid on the other. And then eventually it will form your protein. protein. We'll talk about that. So now the next question is, how does tRNA bind to an amino acid? Well, there actually has its own little enzyme and process to do this. So here we have this enzyme called amino acid tRNA synthase. It's an enzyme. The tRNA kind of look like these broken dynamite sticks without heads. If it has an amino acid, it's going to get a head, in this case a star head. Okay, the star just stands for a specific amino acid. We don't know which one. So you have an amino acid tRNA synthase, the enzyme. And notice we have A and then 3 P's. What molecule is that?
Adenotriphosphate.
Yeah, adenosine
Wow.
triphosphate, or ATP for short. So what's going to happen is an amino acid will latch onto it, and we have ATP. ATP is used, so now we have 2 phosphates and AMP for adenosine monophosphate. Notice that it kind of undergoes a conformational change just a little bit, kind of opens up a little bit. So the dynamite part, which is the tRNA, comes in, and then a bond will form between the amino acid and the tRNA. And now we have something carrying an amino acid. So a tRNA without an amino acid is just called a tRNA. But when it is bonded with an amino acid, we actually call it an amino acid tRNA. So it's loaded. It's got an amino acid. Here's the enzyme that's green, the amino acid tRNA synthase. So notice that we call this the amino acid tRNA. Enzymes are usually named for what they do. In this case, synthase, which means to make. So they make amino acid tRNAs. There is a different amino acid tRNA synthase for each 20 amino acids, because they're very specific as to what amino acid can bind to the top. So it's kind of cool. No, they are not. So the series of steps, just one more time. This one actually shows the conformational change of the enzyme. But we have an active site on the amino acid tRNA synthase, where ATP and an amino acid will bind to it. And then 2 phosphates get cleaved off, undergoes a conformational change. So here it's, I don't know, fat. I don't know how to describe that. But all of a sudden we have an opening, a mouth, if you will, that can fit a tRNA. And then the tRNA comes in and binds to it, forming a bond, and now we have an amino acid tRNA that can go to a ribosome. So the ribosome is actually made up of 2 subunits, a large subunit and a small subunit. And inside the ribosome, there are 3 tRNA binding sites. The first one is called the A site, which I always think of acceptor. This is where the tRNA will first come into. And then the ribosome is going to move over, and that tRNA will go to the P site for the formation of peptide bonds. So this is the middle site, P for, this is where the peptide bond will form. And then it has an exit chamber where the tRNA, after it's given up its amino acid, is ejected or exited. So I have a diagram that shows the 3 sites here. Here's the acceptor site. Comes in, it will latch on, the ribosome will move over, and then now it's in the P site, and another tRNA will come in, and a peptide bond will form between the 2. And then notice the E site is significantly smaller than the A and the P site. Well, the reason why it's smaller is tRNA doesn't fit in this chamber, so it literally gets ejected. Oh, I should go back. So here's the, and I forgot to say, this is the large subunit at the top and the small subunit, and then they come together. So the small subunit is the part that's kind of reading the mRNA, and the large subunit is responsible for forming the peptide bonds. There is a 3-step process to it, and these are the words that come in, but it says amino acid tRNA. So a tRNA carrying an amino acid comes into the A site. The anticodon bonds to the codon in the mRNA. Then the ribosome will move over. Another tRNA will come into the A site, and a peptide bond will form between the amino acids that are in the P site and the A site. And then it will move over, and then the tRNA that lost that amino acid will be ejected. And it just keeps moving down the line. So a diagram, and then I'll actually do it on a slide that I've created. So, A site, P site, E site. Notice we've already started our amino acid chain. So the next step is it's moved over. This tRNA comes in. The anticodon reads the codon on the mRNA. A peptide bond will form between 3 and 4. The ribosome will move over, and then tRNA 3 will be ejected. And all of these amino acids will be stringed to this tRNA, which is now in the P site. So I know that didn't make any sense, so I'll actually do it here.